Good morning, everyone. Today we investigated how alcohol affects plant growth, specifically radish seeds, as a model for understanding biological development. I'm Danica Balila.
I'm Tobias Batista.
I'm Evelyn Chen.
I'm Jessica Green.
And I'm Vex Monsanto.
I'm going to introduce our background for this topic, and followed by that, we'll touch base with methods, results, future study, and conclusion. Beginning with background, we initially decided to collect data regarding the prevalence of substance use among adolescents at our school. However, there were many confounding variables that influenced the study negatively. This guided us to shift our focus more towards an experiment focused on the direct effects of substance use. Due to ethical reasons, we could not directly test these effects. So our study shifted to testing the effects of substances on radish seeds, as they are similar to animal cells. Radish seeds allow for quick and visible growth, providing a visual of development and the effects of alcohols on them, and their similarities allowed us to overcome our ethical dilemma. Through research, we found that alcohol consumption disrupts growth. The image above displays a cell cycle consisting of the four major stages: Gap 1, synthesis; Gap 2, and mitosis. Alcohol can disrupt the most susceptible stage, the G1 phase. Given that alcohol disrupts cell growth and division, primarily during critical stages of development such as puberty, we found that this was relevant to our initial focus. Germinating radish seed cells have many similarities to human stem cells, as they both have the unique ability to differentiate. All in all, we knew that demonstrating the negative effects of alcohol on these radish cells could indicate the potential negative effects alcohol may have on the growth and development of adolescents.
So, how could we show these effects? First, we needed to decide which alcohols we would test. Research revealed that whiskey, tequila, and cognac were commonly used alcohols, so it made sense to test these on our radish seeds. We set up our seeds on paper towels and Ziploc bags in a grid-like arrangement with 35 seeds per bag. This setup allowed each test group to be grown under regulated conditions and allowed for easy data collection. The one difference between each bag was the 20 milliliters of solution that soaked the paper towel. The solutions we used had concentrations of each alcohol mixed with distilled water. We settled on the solutions having an alcohol content of 0.25%, 0.5%, and 0.75%. These alcohol contents were chosen after many trials with higher alcohol percentages that resulted in no growth. After we figured out how to get the radish seeds to grow, we determined the time frame for collecting data was best on days 4 and 6 because there was adequate growth to start collecting the data, but this was a short enough time frame that allowed for easy replication of the experiment. To measure this growth, we used a ruler to determine both the stem and taproot lengths. We defined the taproot as the section where the secondary roots were visible, and we described the stem as being the beginning of the taproot to the opposite side of the germinating seed.
Next, let's move on to the results section in which we'll overview the process of our data analysis that led us to our conclusion. In this first bar graph, we began by focusing on tequila across all concentrations, and as expected, the higher the concentration, the less observable the growth would be. As seen in the graph above, we focused on taproot length, and as the arrow implies, there was an overall negative or downward trend. And because taproot had the biggest differences, for this analysis, we decided to focus on taproot. This next graph, with all of this in mind, compares the different types of alcohol at the 0.75 dilution because, as previously discussed, the higher the concentration, the better the indicator of which type of alcohol had the greatest impact on germination. So on day 4, when comparing the average taproot length of growth, tequila is much less than the other averages, and the same trend is seen on day 6. Lastly, we have a cross analysis between each sample, as seen in the box and whisker plot of radish tap seed length on day 4. When comparing the standard error bar of each solution, there is a noticeable difference when viewing tequila standard error bar as highlighted. Due to its short length, there was little to no variation in comparison to the wide variety experienced by the other solutions, supporting that tequila inhibited the most growth.
So for future studies, we want to study the further effects of plant growth under nicotine and marijuana, and not just alcohol. So these substances are really popular amongst teens as well, and they are known to cause body and brain developmental issues. And using these substances on plants might reflect their impacts on critical development in teens. And so for future studies, we'd probably use plants such as Dale crest because they grow fast and they're commonly used in laboratories. And they also have a fully mapped genome, which means that we could study the effects of things more than stem root and taproot length.
To conclude, after collecting our data, we found that each alcohol concentration with 0.75% had the most impact. The p-value for Gramineer was 0.035, for whiskey it was 0.042, and for tequila it was 0.023. Since the values are below the significance level of 0.05, it indicates that the null hypothesis could be rejected. Alcohol can have an interference of biological growth by damaging the cells, ruining growth hormones, and nutrients not being able to be absorbed. With this, it can weaken the bones and muscles as it develops over time and can have an effect on unborn babies. Overall, alcohol can have an impact on your stunt growth, childhood, adolescence, and pregnancy. Thank you for listening.